All right, so in this video, we're going to look at resting membrane potential. This is a big thing in how the nervous system conducts impulses. So, excuse me, the cell membrane itself is actually electrically charged, which we refer to as it being polarized. Again, we've talked about polar in a bunch of different times. Polar meaning one side's positive, one side's negative. In this, what we're going to see is, like, if this is the cell membrane and this is the inside here, and again, I hate the fact that I can't, we're going to find that there's positive ions we're going to see outside and positive ions on the inside. And it just happens to be that because there's more positive on the outside, it ends up causing there to be a difference in charges, and the membrane itself becomes negative because of this difference in amounts of the charges from one side to the other. And I'm going to explain it a little bit more as we go on, but that's just the gist of things. So polarized, again, means I've got these opposing charges, basically. And so, as it says, so that the inside of the membrane is negatively charged with respect to the outside. As a result of unequal distribution of ions on the inside compared to the outside of the membrane. And this is how impulses, these little messages, are sent back and forth on neurons. And again, it's the same thing that happens in the muscle fiber. We don't get into it as much, but it is a change in the cell membrane potential that causes the sarcolemma, the outside membrane, to change its electrical characteristics, and it basically becomes electrified. So the two ones that you really need to pay attention to, sodium and potassium, these are the ones that we really look at in here, sodium and potassium. Again, if this is the cell membrane and this, and I'm just going to draw an arrow instead of that, and I'll say this is, well, I better put the thing inside and this is outside. What's going to happen is sodium is on the outside of the cell and potassium is on the inside of the cell. And there's a lot more sodium than there is potassium. And so that's what causes the difference in charges. But you have to remember that it is potassium on the inside and it is sodium on the outside under normal things, because that's what's going to cause this charge, this electrical wave to go down the axon. It's going to be the percentage, because we're going to have things we're going to see in just a minute. Sodium or potassium wants to leak out and sodium wants to leak in, and it's this sodium-potassium pump that keeps this cell membrane potential, this electrical charge, where it needs to be. So the sodium-potassium pump is going to take these sodium ions and push them back out of the cell. It's going to grab the potassium ions that have leaked out of the cell and bring them back in. So it's this little Ferris wheel that's kind of spinning around and taking the potassium and putting it back in, and taking the sodium and kicking it out. It's the bouncer of the club called axon. And all along here, we're going to have what are called ion channels. Now, these ion channels we kind of looked at. This is a picture of them. We looked at this way back in the beginning when we talked about how things move in and out of cells. They help regulate how the ions move in and out. Now, we're going to look at a couple of different types of them. Now, on the axon, every one of them is electrically controlled. But on the cell body and the dendrites, they end up being, and I don't know why I drew that arrow, but they end up being controlled by multiple different things. The ones on the if I was going to draw a cell here and have all these as axon or dendrites, and then have the one axon, looks like some of them got kind of scared, but and if I drew a line here, all of the ion channels on this side are electrical. Everything else on this side is going to have different things. Pressure can open them, chemicals can open them. So when we're talking about how our body controls blood calcium levels. We talked about this in the skeletal system. These little dendrites are going to be sitting out here monitoring the calcium levels in our blood, and if the calcium gets too high or too low, the concentration of that's going to open these channels and cause an electrical impulse to go back to the cell body. So, but the one that we're really going to look at most in this chapter is this electrical, how the electrical ones work on the axon. So we have this, what we call resting membrane potential, which is simply the normal charge of a neuron cell membrane. So again, I've got this cell membrane, and I'll draw it on this side. So potassium is inside here, right? It's all in. And then sodium is all around outside. And again, forgive me trying to draw things with the little that little pad on my laptop is not easy. So sodium is surrounding it all over. And since there is far more sodium than there is potassium, it ends up being where even though all of these are positive ions, because there's more positive ions outside than inside, it sets across a gradient, it's called, and it ends up making it to where the cell membrane itself becomes negative. So imagine that we're in a room, and we're, all of us cool science people are in here, and there's like a whole bunch of English majors outside in the hallway, but there's only 20 of us in the room, and there's 100 of them in the hallway. There ends up being, if we're all positive, it ends up being where there's a difference in the amount. So if I have 100 positive people on the outside of the room and only 20 on the inside of the room, there's a difference of 80. And so it would be the wall would end up being a negative 80. That's because of the difference. And that's kind of how it works with this. Now, because in here, instead of 80, the resting membrane potential ends up being 70. So it is down here, it talks about it is a negative 70 millivolt is the charge of the cell membranes. And that's why it is referred to as being polarized. So that's how it is when it's normal. And in order to send a signal, I've got to disrupt that negative 70 millivolt charge. And it's always kind of set and made sure it's in the same spot, reset by the sodium-potassium pump. Now, these next few slides kind of are weird. They kind of talk about how this happens. It's in a hypothetical neuron before the membrane potential is established. That's crazy. It talks about what's happening, how potassium would leak out. And again, this is all hypothetical. But remember, we're going to have this little thing here. This does happen as far as potassium comes out, sodium tries to come in, and so this sodium-potassium pump is going to grab this potassium that tried to get out, and it's going to pull it back in. It's going to grab this sodium that tried to sneak in, and it's going to throw it out. And so we have this little wall, and this is correct. Again, it's the hypothetical. It's trying to show you how it is set up, but then we end up having this little difference in charges from one side to the other, and it again is measured as negative 70 millivolts. That is the resting potential. And again, this is talking about how the sodium-potassium pump is the thing that keeps it set as it is. So again, remember, I'm going to look at it on this next one. So I'm just going to draw on this blank slide to just see if hopefully it'll make sense. So on this slide, if I just kind of draw this little, we'll say this is an axon, and I've got potassium in here. And it is, potassium's all over. And I don't know. So we got 3, we got 6 there. And then out here, this is the interstitial fluid. This is the fluid that's around things. We're just going to have so much more and of sodium. And again, these are both positive ions. Remember, positive ions are called cations. And again, you can remember cat people are positive, even though I don't always agree with that. But I'll keep that to myself. So we've got all of these sodium ions. Na, na, na. I think that's a song. But anyway, what ends up happening is, again, because there is, let's say there's 6 positive on this side, and on this side there's 7, and then there's another 13. So it's like there's 6 and 13 here. Now in this hypothetical situation, it ends up being when there's 7 more. So there would be a -7 charge at this membrane because of the difference that's surrounding it. And if I was going to do it in the correct order, which I wouldn't be able to do with all these, with the way I'm drawing it here, because of the difference, it actually ends up being where the difference is 70. So that is why we end up having this -70 millivolt resting membrane potential at the membrane. And so when we're going to look at in a little bit how an electrical impulse goes down the axon, and it's going to be because we're going to have these gates that open, and sodium's going to rush in and potassium's going to rush out, and we're going to change this -70 to form this little wave that goes all the way down the axon to where it's going to end, and it ends in something called a synapse. We kind of looked at those when we were in the muscular chapter last time. We called it a neuromuscular junction. All it is a synapse where the axon, that wave goes down to the axonal terminal, and it causes a release of something called a neurotransmitter. And then whatever cell, it's usually a neuron or whatever is on the other side, that's going to bind to receptors and cause that membrane potential to change. And that's how that kind of works. I hope that makes sense.